Well, thank you very much, Lisa, for inviting me to speak today. I'm excited to share with the audience my work related to feeding and salivary analysis in the newborn. I have no financial disclosures or conflicts of interest. My objectives today are to identify the challenges associated with assessing oral feeding skills in the newborn and to assess current tools available for determining readiness to feed in this population, along with their limitations. I hope by the end of the talk, you will understand the benefits of saliva as a non-invasive diagnostic biofluid, as well as its potential into determining readiness to feed in the neonate and diagnosing other feeding problems in infancy. But my real objective for the talk today is to challenge our current thinking about oral feeding assessment and treatment strategies in the newborn. So let's start with a question. How often are you confronted with a newborn who cannot successfully feed? I imagine in this group, it's quite common. And as a group, as a whole, we're really good at discerning between infants who can and cannot feed. They can either take the bottle or go to breast, or they can't. But this is very distinct from understanding if a baby is ready to feed. So another way to think about this is how many times have we actually known why that infant cannot orally feed? Typically, we make up anecdotes based on our experience about all the different feeding problems we see in the NICU. For example, these big, large babies born to infants of diabetic mothers who are in the NICU because they refuse to take oral feeds, we tell each other they're pokey feeders. Infants with persistent pulmonary hypertension who were born at term but now won't eat, well, they won't eat because they were so sick after birth. Infants with neonatal abstinence syndrome or NAS, those babies won't eat or they eat funny because they're not captured in their withdrawal. Boys don't eat well because they're boys and we blame them on their sex. And then of course we have the 35-week infant who the nurse says fed completely fine at noon, ate the whole bottle, but will not wake up at 3 for the next feed because it's been determined the baby must be too tired. I would say that all of these are anecdotes, and they're anecdotes because feeding's complicated. And we know that oral feeding competency relies upon the maturation and coordination of suck, swallow, and breathe. There is no doubt that if a baby cannot do those 3 tasks simultaneously, they're not going to eat. However, feeding is more complicated than that. It's driven by the senses, sense of smell, taste, touch, hearing, and vision are all important for how a baby is able to feed. So too is the gut-brain axis. You can't just know how to feed. You have to know you're supposed to feed. You have to know you're hungry in order to wake up to do so. And of course, there's neurodevelopment, which is occurring rapidly in this population. And some babies reach their milestones before others. We also know that oral feeding and oral feeding strategies are not one-size-fits-all models. Babies learn to feed at different rates, they have different challenges, and they are not a homogeneous population. In 2018, a multi-center retrospective analysis of a prospective cohort of moderately preterm infants that were admitted to the National Institutes of Child Health and Human Development Neonatal Research, or NRN hospitals, were analyzed. Their primary outcomes in this study was post-menstrual age at full oral feeding and at discharge home. And the group looked at over 6,000 infants that were born moderately premature between 29 and 33 weeks between January 2012 and November 2013. And they looked at a variety of factors that influence feeding. And here I present a table from the article of factors that prohibited feeding, that made a baby take longer to feed or less time to feed. And it's listed here, age at when they were first started, what their weight was, what birth age they were, whether they had respiratory issues as surfactant exposure, had a PDA that was treated. But what I want to draw your attention to is the following. The first is that you were less likely to feed if you were male. So anecdotally, we've seen this, boys will be boys, but this was one of the first studies really to look at a large cohort and said, you know what, boys don't feed the same as girls, and they're not feeding on time like girls. And we also see this anecdotally, but this study confirmed it in this large cohort, was that if you were of the Black race, you were going to learn to feed sooner. So what we see here is that this is not the same group of babies just because they were born at a certain age. There are variables that impact that. And when it comes to male, these data are increasingly getting stronger and stronger. We actually know now, based on studies coming out, and this one's even later at 2006, it said, when we look at fetal ultrasound as early as 15 weeks, we can see that oral motor and upper airway skills are emerging earlier in females. Again, reiterating the fact that sex alone is a determinant of feeding outcome. So how do we determine when we go to the bedside and we know the baby can't eat, what their sensory integration status is? Or how do we assess their gut-brain access? How do we assess oral motor skills? How do we know the baby is neurodevelopmentally ready to begin oral feeding? And of course, the answer for us is, well, cue-based feeding, infant-driven feeding assessment. And this is taken from Ludwig and Weitzman from their 2007 paper that we use as one of our gold standards for infant-driven feeding, where we ask the question, if you are born or you're correct to greater than 32 weeks, PCA or post-conceptional age, and you are on minimal respiratory support or breathing room air and full enteral nutrition, do you show readiness cues to feed? And if you do, and you consistently show those, you're allowed to feed at least once per shift. And if you aren't showing that, then you'll continue to wait until you meet those criteria. At my hospital, at Tufts Children's Hospital, we have this integrated into our daily vitals. So the nurses are doing this all the time once a baby reaches 32 weeks post-conceptional age. And in essence, what these tools are allowing us to do are answer those questions that I just asked. These are surrogates for those important developmental questions. For example, waking to feed is a surrogate for maturing gut-brain access. What you're looking for is the baby hungry? Did the baby know that they were supposed to wake up and feed? Sucking on a pacifier is a surrogate for oral motor readiness, their ability to suck and suck vigorously. But it begs the question, can we actually do better? And this has been looked at in 2 Cochrane reviews, both in 2012 and 2016, where they undertook a review to assess the effectiveness of oral feeding assessment tools to reduce the length of stay and shorten time to establish full oral feeds. And what they concluded in both reviews was that there were no studies that met inclusion criteria, concluding there is currently no evidence to inform clinical practice, and that research is needed in this area to develop an instrument to assess feeding readiness in the preterm infant population. So where do we stand? Well, infant-driven feeding is the current gold standard. It's what we use. It's how we assess babies' readiness. And there are numerous devices on the market to improve feeding outcomes. Some of these focus on sensory integration or oral feeding training, training the mouth how to eat. But it begs the question, when we go to the bedside, which infant needs which intervention and when do they need it? If we start with oral motor maturation, there are 2 devices at least on the market, one being the N-Trainer. This is a somatosensory training machine where it simulates the normal suck breathe pattern for these babies, but it's not associated with a bottle. There is no feeding going on simultaneously. Although you can use the intervention when a baby is receiving an NG feed or a nasogastric feed to help the baby coordinate the 2 together, feeding in the stomach with working on oral motor control simultaneously. In contrast, there's the N-Font, and this is an oral motor assessment tool that happens simultaneously with feeding. So it adapts to nearly any bottle, and while the baby's eating, you can assess their oral motor skills. We also have available to us sensory integration tools. One, of course, is the pacifier activated music player, also known as a PAM, where music is played as the baby is sucking to reinforce the appropriate suck. And of course, in 2014, Chorner et al. In pediatrics substituted the music for the mother's voice to show improvement in oral feeding outcomes. My own labs actually looked at breast milk olfactory stimulation. This is a baby that was in a trial that we did that I'll discuss briefly, where we would stimulate the baby with the smell of breast milk while they were getting an NG feed. And we did it on the cotton swab that little baby is holding. What we did was we enrolled infants born between 28 and 33 and 6/7 weeks gestation. There were 36 total, and they were randomized to either receive mother's own milk or mom, or water being a sham stimulus during the learning process of oral feeding. We recorded clinical and feeding outcomes, and we did statistical analyses to look at the effect of stimulation with mom's own milk on feeding outcomes stratified by age and sex. Here we can see a schematic of the study design. Most babies had enteral nutrition initiated by day of life 0-5, and we initiated olfactory stimulation on day of life 1 to 8. And we continued that at least once per day by 4 or greater days per week throughout the time that the baby started to receive enteral nutrition until they received full oral feeds. And what we found was that there was actually no significant differences in the whole group between sham infants compared to mother's own breast milk infants in mean postmenstrual age at full oral feeds. But if you just looked at infants that were born less than 31 weeks gestation who received mom's own breast milk stimulation, they did learn to feed sooner than controls. And this suggests that perhaps there's even an ideal developmental window for the intervention. Here, we saw no sex difference in response to olfactory stimulation. So in order to fully understand which baby needs which intervention when, we must be able to assess their development in real time. And this is a big question. And I asked this question years ago. How do you do this in a highly vulnerable population? And of course, the answer was saliva. So why saliva? Well, for those of us who work with newborns, it's pretty obvious. It has several benefits over other body fluids. It's non-invasive, right? I can collect it any time I want by simply suctioning their mouth, and I will not hurt them. And importantly, parents say yes to enrolling their infants into studies that only involve saliva and not blood. Saliva, though, is a direct filtrate of blood. It contains almost everything you can see in blood, including electrolytes and cells and glucose. It has proteins, hormones, enzymes, drugs, immunoglobulins. All can be measured in saliva. It's a rich source of microorganisms. And for me, it contained an enormous amount of genetic material, both DNA and RNA. So my laboratory has used saliva to objectively monitor multiple developmental systems simultaneously in babies, including their oral motor control and facial development, their sensory integration, including olfactory, vision, hearing, and taste, hunger signaling, neurodevelopment, and gastrointestinal development. My goal is to develop a diagnostic assay to assess an infant's readiness to orally feed, identify the developmental delays limiting oral feeding success, and personalize our approach to treatment strategies based upon an individual's salivary profile. So for over a decade, my laboratory has attempted to understand molecular mechanisms and developmental pathways involved in oral feeding, and we've done this at many levels. We've done it at a genomic level, and that's our DNA, looking at mutations within specific genes. And I'll give you an example of that. I'm going to spend most of my time talking about my transcriptomic, or RNA. That's what's read off the DNA in real time, and it tells me about the developmental status of the infant in that moment. And I've also done some proteomic work, which are proteins, and that's the real mechanism of action. We've done some work in saliva, but for the sake of this talk, I will not be discussing my proteomic work. So let's begin with the genomics of feeding. And I'd like to share a story with you that occurred with my collaborator, Dr. Emily Zimmerman at Northeastern University. Dr. Zimmerman is a speech pathologist, and she came to me several years ago and was very interested in this gene FOXP2. And as a speech pathologist, she was aware that FOXP2 was the first gene to be implicated in a developmental disorder of speech and language. And it was based on molecular studies of 15 individuals in what was identified as the KE family who suffered from speech language disorders. And Emily hypothesized, well, this gene might be very important for feeding because of the shared oral motor groups that are needed for both developmental outcomes, feeding and speech. So we were looking at FOXP2 in saliva and running studies that we were able to publish. And while we were doing that, I was emailed by a woman. And she wrote to me and said, I came across your name while researching my son's recent diagnosis. My son was born via C-section at exactly 35 weeks because I had preeclampsia. He suffered no trauma during pregnancy or labor. He was in the NICU for 42 days for suck, swallow, breathe. We tried breastfeeding, formula, thickened formula, different nipple sizes, spot feeding, etc. We had no explanation for why he couldn't coordinate suck, swallow, breathe. He underwent an ultrasound of both his brain and his heart, and he had an MRI. All findings were normal or non-significant. And after failing a swallow test with flying colors, he had a G2 placed. It was only then, in an effort to find the cause of the issue, his neonatologist ordered a microarray and chromosomal analysis, where it was revealed that he had approximately a 9 kilobase loss within the chromosome band of 7q31.1 that contains exon 2 of FOXP2 gene. So what this represented was we had the first case ever that we knew about that linked a genomic disorder with poor feeding in the neonatal period. And I would ask all of you, how many times do you think about a non-dysmorphic baby having a genetic disorder just because they can't feed? And I would argue I certainly didn't. I didn't do it enough, and we probably missed a lot of these babies. And at the time, to our knowledge, this was the very first case linking a deletion in FOXP2 to oral feeding impairment in the newborn until this past month. In December of 2020, we received an email from the Netherlands. And in it, the doctor wrote, with great interest, I've read your case report about an infant with feeding difficulties and a FOXP2 deletion. I'm a pediatrics resident in Wilhelmina Children's Hospital in Utrecht in the Netherlands. And a patient of mine also has a FOXP2 deletion. This 1-year-old boy has feeding difficulties very similar to the case in your article. I would argue that there are probably dozens and dozens and dozens of babies, many of whom we've all probably taken care of, that have FOXP2 deletions that we've never searched for, that we've never recognized, that we've never understood. But these cases serve as important reminders to consider genomic testing in infants who cannot successfully feed by term, particularly those who lack related pathology that may limit their feeding success. So we know genomics may be important, but let's move on to transcriptomics. And that is that real-time expression I talked to you about, that RNA that's being read off the DNA to tell us what can the baby do today. For years, I looked at the saliva of successful versus unsuccessful oral feeders at an RNA level in order to gain an understanding of the developmental status of that newborn in the moment. All of this was done in saliva, and I used various scientific platforms to do it. And what I found was, sure enough, if I took a drop of saliva, I could simultaneously detect genes involved in all of these systems: oral motor, GI, sensory input, neurodevelopment, and something that always piqued my interest, which was feeding behavior. So when we first started this, identifying feeding behavior, quote, pathways in newborns was pretty novel. Most people weren't focused here. They were really focused on oral motor control. And there was very limited qualitative data assessing hunger signaling in these babies, satiety, how they were being regulated for food intake, and how the hypothalamus was regulating feeding behavior. And the important role of these biomarkers involved in feeding behavior made perfect biological sense for us, because I want you to think about something. On average, a newborn gains 200% of his birth weight by 1 year of age, 200%. So if they're born at 8 pounds, by 6 months, they should weigh 16. And by a year, they should weigh around 24. A preterm infant could gain 300, 400, 500% of his birth weight in that first year of life. And in order to do that, a newborn will consume 80 to 150 kcals per kilo per day. And to put that in perspective, caloric intake of a newborn is equivalent to an adult taking in 7,000 to 10,000 kcals a day. This here is only 3,800 calories. So I want you to think about that in context. And remember, babies don't do anything, right? They sleep, they eat, they poop. That's about it. They cry. So they're not running marathons and they're consuming that amount of calories. So one of our very first successful transcriptomic or RNA hits was related to oral feeding in the newborn, and it was involved in hunger signaling and a maturing gut-brain axis, and it was a gene known as MPY2R. MPY2R modulates feeding. It's a little counterintuitive because when it's turned on, you feel full. You have satiety, and when it's turned off, you get very hungry, and it does fluctuate throughout the day, and that's how we know now it's time to eat, now it's time to stop eating. But we knew that this was an important gene to allow you to go from this at birth to this within 1 year of age. So in neonatal saliva, we looked at MPY2R, and it turns out on the assay we were using, we could use it in a binary fashion, meaning if it was on, great, but if it wasn't, it was also informative, and it was just an on-off switch. And what we found in this initial work was that if we could amplify the gene, if we could detect it, we could predict with 95% positive predictive value that that infant could not sustain full oral feeds. They weren't ready to do it. They did not have a mature gut-brain axis driving feeding behavior. But the negative predictive value of the assay was only 27%. In other words, you can be hungry and you may have that axis that's mature, but you still have no idea how to do it. Okay. And this led to our complexities. So I needed to do more research, and I needed to develop a diverse gene panel for the prediction of oral feeding readiness. So I went back to work. And I actually looked at over 400 salivary samples to identify this panel I'm going to show you today. And this panel is 5 genes highlighted here that represent a diverse range of biological functions required for successful oral feeding. 2 are involved in hunger signaling. Here's our NPY2R, and we also looked at AMP-K. 2 are involved in sensory integration, plexin A1 and NPH-P4. And 1 is involved in facial development, Wnt3. So let's talk about these genes. I told you we can look at the genes like an on-off switch, which makes the readout really easy. So a successful oral feeder, if I look at all 5 genes, will have turned on AMP-K, which regulates whole body energy balance. An activation of the gene in the hypothalamus induces feeding and weight gain. They're also a successful feeder if they turned on plexin A1, which controls axon guidance, and increased expression is known to occur in mature rather than developing olfactory sensory neurons. But these same babies simultaneously have to have turned off MPY2R, because again, down regulation of this gene induces hyperphagia. They need to have turned off Wnt3 because this is an embryological gene involved in lip, palate, and tooth formation. So that should be pretty complete if you're ready to feed. And then turn off NPH-P4, which is involved in retinal development and visual behavior. And when we looked across our group of our 400 babies who could successfully feed, here are the genes with the age and sex, sensitivity and specificity of each gene individually being able to predict oral feeding success. Here's our positive, negative predictive value, odds ratios with confidence intervals, and our p-value. And what we found was that there really is no magic bullet anywhere. But I want to call your attention to age. Age here mattered. The older you were, the more likely you were to feed. And sex mattered too. We could see it at a molecular level. As a matter of fact, you were much more likely to feed if you were a female than if you were a male. In terms of sensitivity, AMP-K is pretty sensitive at the expense of specificity. Wnt3 is pretty specific at the expense of sensitivity, but none of these alone are going to tell us the story. because there is no single magic biomarker for determining readiness to feed. It's complicated. So we asked the question, well, if I combine all 5 of those biomarkers together into a predictive model, how accurate are we? And we weren't bad. So what I'm showing you here is all 5 genes together in their correct on-off configuration at predicting oral feeding readiness. And this is what's known as an area under the receiver operating characteristic curve, where you plot your sensitivity on the y-axis, your 1 minus specificity on your x-axis. And this line here is no better than flipping a coin. So obviously an assay that fell on this line was useless. I could have flipped a coin and done better. But what we see here is that we're not that bad. We're actually close to 80% accurate using this assay to predict oral feeding readiness. There's room to go, but we're not that bad. But the real power of it was here. So let me walk you through this. I'm going to take a 35-week female and a 35-week male. I'm going to list all my genes. These are the genes that would have to be negative or turned off to feed, and that's highlighted in green. Those are those 3 genes. And these are my positive genes, AMP-K and Plexin A1. And if I take all those genes that should be turned off and all those that should be turned on, I can predict that a boy at 35 weeks has a 60% chance of feeding. That girl has a 73% chance of feeding. And here you see the power of sex. Similarly, if all the genes are in the wrong direction, that boy only has a 3% chance of oral feeding, where the girl has 6%, and then every permutation is in between. But what if I just knock out 1? What if I just say their olfaction isn't ready? Well, here I go down to 35%, just that 1 gene. And this panel may suggest that's the baby that needs breast milk stimulation. Not an oral motor training tool, but something that's actually going to engage their sense of smell. So these data analyzed 2 important aspects of feeding maturation. If you wait long enough, infants will feed, and girls are going to do it sooner than boys. But it also laid the foundation to know that you could look at a panel of genes and begin to understand why that infant in your care can't feed and how you can start to gear your treatment strategy individually. But I needed more studies. I still had a knowledge gap. Remember, we're only 78% accurate. There is room to grow. So we needed to understand what else was involved in oral feeding maturation. And so we went for the very first time to a platform known as RNA-seq in hopes of improving our understanding of oral feeding competency, identifying novel pathways we had never seen, and allowing for the development of personalized approaches to improve feeding outcomes. Now, RNA-seq is hypothesis discovery. In other words, unlike approaches that quantify only a select number of genes, RNA-seq is going to allow me to investigate all the genes being expressed at a given time. And I can look at these expression profiles between groups, for example, cases or controls, to identify genes that may be involved or causing a condition. It allows the investigator to generate a hypothesis of the pathophysiology. So we performed RNA-seq on saliva samples collected from both successful and unsuccessful oral feeders. And we tried to do everything we could to match the cohorts so that that wasn't infected by the gene expression. So we matched them by the gestational age they were born, the post-conceptional age at which we got saliva, their sex, and even their ethnicity. And we performed comparative and system biology analyses on differentially expressed genes between both successful and unsuccessful oral feeders and those males and females. And this is what we found. When we did this, there were 63 genes that were differentially expressed between feeders and non-feeders, and they cluster differently. So you can see our feeders are in yellow here. These are each individual babies, and our non-feeders are in blue. This is a heat map. So those were all those genes that we could see, and we could see that the non-feeders look different than the feeders, but they're not completely separated. We've got a few outliers here, a few that cluster in the middle. So we could pick up these 63 genes, we could see visually how they were clustering, and then we can delve right into it. And when we did that, those genes were involved in nervous system, including memory and myelination. They were involved in structural maturation, specifically of the palate, and they again were involved in sensory integration of vision and smell. When we looked into the nervous system developmental pathway, genes mapping to cranial nerve development and sensory integration were the most statistically significant differentially expressed. This include cranial nerve 3 and 4, as well as formation of retinal ganglion cells and abnormality of the aqueous humor in the eye. We also saw size of olfactory bulb related to cranial nerve 1. So again, olfactory and olfaction is a huge component of oral feeding readiness. When we looked at other areas of biological relevance, we could see palatal shelf formation, maturation of the circadian rhythms, again, sort of knowing when you should be eating, abnormal morphology of hindgut and mesenchyme, and development of the abdomen. But sex absolutely matters. When we did an independent analysis of males and females to look at unique differences in oral feeding between the sexes, we found that there were 88 genes differentially expressed amongst the girls, and there were 77 genes differentially expressed amongst the boys, and there wasn't a single gene that overlapped. They were completely different. So let's look at those males. Here you see a huge split. They're clearly splitting of the males who could and couldn't feed. You can see this graphically. Here again is your heat map. There is no overlap between our feeders and non-feeders, and here are all those genes. And again, we asked the computer, what are all those genes that are differentially expressed? And what we saw with the males was nervous system, hair and skin, connective tissue, a lot of development. But for us, the money was in nervous system. You can see the p-values and the number of genes that fell within each category is also listed here. So what was interesting for us was that for the very first time, we could see disruption in memory and learning, and it was only ever seen in the males, meaning they couldn't remember how to feed. That was our hypothesis. They were having trouble. So for those babies, something like an N trainer or an N font could be extremely important to remind them, this is how you do it. This is how you do it. When you delve deep, you could see exactly where in the hippocampus that these areas were affected. They also were affected by myelination and myelin sheath, and then areas of the brain were also highlighted. So when we got to the females, there were 88 genes that were differentially expressed. These two are close call, but otherwise there's a better, clearer split here as well. Again, here's our heat map. These are our genes, and we asked the computer, what are these genes doing? And what they told us was not quite as exciting. Here we can see some blood development, immune cell trafficking, humoral immune response, lymphoid, a lot of blood work here. But we went here. We wanted to see what was going on in the digestive system of our females that was separating feeding. And here's where the palate came in. So for this group, we could see here, unlike the boys that had nervous system immaturity, the girls seemed to have more of a facial and an abdominal immaturity, including formation of the hard palate, secondary palate, tooth development, and the incisor, and then the villi of the GI tract. So these data highlighted the important impact of sex on acquisition of oral feeding. We know there are differences there, particularly between males and females in terms of our outcomes, complications, and milestones, but now we can see that on a molecular level that hopefully will help elucidate how we better treat these babies at the bedside. So where are we going from here? Well, we're in year 5 of an NIH-funded multi-center clinical trial to further test the predictive accuracy of what we call Nourish. That's our platform that we developed in a cohort of infants born less than 29 weeks gestation. It's one of the first neonatal salivary diagnostic clinical trials ever to be done. I do this in collaboration with my PI, Dr. Stephen Barlow. And here what we do is we randomize babies to receive the N-trainer feeding device, and we collect saliva throughout the treatment and learning process of oral feeding. This is our very first attempt to understand how these genes change over time in response to a treatment. This will be a big year for us. We anticipate having several papers submitted and hopefully published within the next year regarding this trial. So I want to end just by sharing some work that we're doing with feeding to make you think about all the types of babies you deal with in the unit who are having feeding difficulties. So I've talked largely here about premature babies, but can we apply these same scientific approaches to the spectrum of infants in our care who have feeding difficulties? Infants of diabetic mothers, those with persistent pulmonary hypertension, and those with neonatal abstinence syndrome. I'm going to focus the remaining talk on NAS. So we are in a crisis in our country where every 25 minutes a baby is born with NAS in our nation. And oral feeding absolutely affects these babies as well. Infants exposed to in utero to illicit drugs exhibit aberrant feeding behavior. First, they often may have an uncoordinated or an ineffective feeding phenotype, and this is often followed by hyperphagia. Whereas we talked about before, a term infant may consume 80 to 120 kcals. Excuse me, infants with NAS can consume 150 to 220 kcals a day or more. And we see this all the time. And we have said that that excessive caloric intake has been attributed to increased caloric demand due to withdrawal syndromes. That's another anecdote we say, we make that up. So this is a picture of the arcuate nucleus of the hypothalamus. And I only bring it up because the arcuate nucleus derives when we don't eat and when we want to eat. And I want to focus right here. I've talked a lot about the hypothalamus and feeding. Here's MPY. Here's your NPY2R receptor. Here's your leptin. Here's where we're driving hunger. But you know what it sits right next to? This is our mu receptor, our opioid receptor. This is where Narcan works. Okay. There's this is our cocaine and amphetamine receptor. It's right there. It's right next to our feeding center in our brain. So our reward and our feeding are intricately linked in our brain. And so we ask the question, are they overeating because they're burning too many calories? Or are they overeating because when they were developing, this close proximity to food and drug got disrupted? And now food is replacing the narcotics that we are withdrawing them off. And now food is becoming their drug. So in utero exposure to illicit drugs results in imbalance between our energy-driven and our reward-driven pathways. And we hypothesize that salivary gene expression profiles will differ between cases, infants with illicit drug exposure, and control, sex and age matched infants, as well as between male and females. And that we could use salivary gene expression profiles to correlate with total intake in infants with NAS. Again, this is what our hypothesis was, that food and drugs end up along the same, converge along the same pathway of reward. We like the taste of chocolate chip cookies, it rewards us, we want more, and drugs similarly have that effect. So we did a targeted analysis. We went after certain genes, unlike our RNA-seq where we were discovering, we knew what we were interested in. We went after NPY2R again because that was such a strong signal. And we picked up 2 more genes related to feeding. One is POMC, which is involved in hunger signaling with a decreased expression, increases appetite. And the other is the leptin receptor. And then we went after the dopamine 2 receptor, which is a major reward signaler, which is known to have increased expression with drug exposure. This is Dr. Elizabeth Yen. She led this work within my team. This is a prospective case control observational study, and she collected saliva from subjects within 48 hours of birth prior to any treatment for NAS. And she looked at saliva and we ran PCR with proper normalization. To be enrolled, you had to be greater than 34 weeks. Your mother or the baby had to be positive for opioids or cannabinoids, and we matched for sex and gestational age. We excluded infants of diabetic mothers because they have other feeding issues, as we know, and we also excluded women on SSRIs. They were also in that that diagram I showed you, and we know babies can have withdrawal symptoms related to SSRIs, and there were no babies enrolled with anomalies or CNS abnormalities. We worked out of 2 hospitals for this study, and we recruited 103 subjects, but we had 3 that failed at the salivary level. So there were 100 subjects enrolled, 50 cases and 50 controls, and here are our male-female splits. And of the cases, we saw 11 males required treatment, 16 didn't, which is common. Males more often require treatment than females. And here we saw females, 5 of which required treatment, 18 did not. So what we did when we ran those salivary biomarkers, we actually saw no difference in gene expression between cases and controls. They weren't different. And when we looked just at the cases, those that required treatment versus no treatment, there was no difference either. But when we separated by sex, there was the difference. Expression of DRD2 was significantly differentially expressed between male and female case infants, with a higher expression being in males. And that was true if the infant required treatment for NAS or not. So males seemed to be having greater NRD2 expression, and they were more likely to require treatment. When we looked at their intake, actually expression of DRD2 also correlated with volume intake. Infants who had the highest level of DRD2 on days of life 2 and 7 took in the greatest volume. And we also were able to detect here POMC, which is a feeding driver gene again, and that correlated as well. Infants who expressed the highest levels of POMC on day of life 2 took in the greatest volume. So these pilot data suggest that hyperphagia, often seen in infants with NAS, may be due to developmentally disrupted reward signaling pathways. In other words, food becomes their drug, and that sex-specific differences in these pathways may provide a molecular basis for the clinical outcomes we see in infants. Dr. Yan has recently been funded to compare functional and structural MRI studies on newborns exposed to in utero opioids with sex and gestationally age-matched controls. And what she does is she feeds the baby, and they directly go in the MRI machine right after a feed, and we watch how their brains light up with particular interest in the hypothalamus. She's also looking at the role of inflammation in saliva on how opioids affect the developing brain, and hopefully will perform long-term follow-up on infants to assess their development, feeding behavior, and addiction. So the last thing I'll give you a tease on is we're also studying our diabetic mothers. Again, our pokey eaters, some of who require NICU admission and NG feeds despite their large size. And prior studies have suggested that these infants have an immature neurodevelopment and neurodevelopmental system, and that's why they don't eat. But we're asking a different question. We're putting forth an alternative hypothesis that these babies are in an overfed state in utero with increased insulin levels, and that is disrupting their gut-brain axis, their feeding, and fat regulatory genes such as leptin. And in response to these in utero exposures, infants downregulate their appetite in order to normalize their growth curve within the first year of life, which we know they do. Infants born to diabetic mothers may be born large for gestational age, but by the first year of life, the vast majority of them will fall along the normal growth curve. So we are conducting an ongoing prospective observational study. We're comparing term infants born to diabetic and non-diabetic mothers. We look at their body composition, their feeding intake, and salivary profiles. So stay tuned. So feeding's complicated. The developmental complexity required for successful oral feeding has a heterogeneous group of limitations that prohibit oral feeding success, and that requires us to individualize approaches and treatment strategies. And the molecular basis for oral feeding difficulties is informative and relative for both short and long-term outcomes of our infants. Remember, they're not the same. Sensory integration isn't facial development, and it isn't suck, swallow, and breathe. And why an infant cannot feed should inform treatment strategies to improve outcomes, with males appearing to be on a distinct developmental time course compared to their female counterparts. And I hope I've convinced you that saliva is an important tool to gaining this information. So we need to be able to monitor and assess multiple biological systems simultaneously in real time to truly understand the mechanisms underlying oral feeding maturation and oral feeding difficulty. We must recognize the importance of sex on development. It matters. And we can advance genomic platforms can aid caregivers in determining specific areas of delay or disrupted development that may be prohibiting feeding success. We indeed have an important opportunity to individualize care plans and feeding strategies. And with that, I'd just like to acknowledge my laboratory and the Mother Infant Research Institute at Tufts Medical Center, all the families who graciously participate, as well as the nurses and staff in our NICU, and the current and recent funding sources that have made my work possible. And with that, I am happy to answer any questions.
Investigate that. So let's ask some questions. First, I also have to just commend you and thank you for all this research. You know, we all see sometimes on these Facebook posts about, oh, help me, I have this baby, can't figure out why he won't feed. And I think that for our future, this is so exciting because it may present some answers to these questions. So thank you for all your research. Let's try to tackle some of these questions. All right, so first of all, you know, people of course want to know, this is research now, but is it going to be something we can actually use, and when and how? So if you could maybe address that, that's one of the biggest questions we're getting.
Yeah, sure. And it's a great question. And it is still in research. And I mentioned that we're coming to the end of our year 5 trial that I've been doing with Dr. Barlow. And why that's so important is these are thousands of saliva samples that we're testing. And if our accuracy remains, and if we're able to identify areas that may be limiting feeding success in babies, and importantly, watch how the end trainer helped with maturation of babies feeding, we can then take our next step. And for me, that next step is taking all that NIH research and bringing it over to industry to bring it out to all of you. Of course, as a diagnostic, I'm regulated by FDA, which goes through the same trials as a drug, goes through phase 1, 2, 3, 4. So it's a slow, tedious process, no question about it. I'm trying to go as fast as I can. And if you have a hospital that's really interested in this research as we move forward and you want to be part of studies, you can certainly reach out to me. I know my own speech pathologists, OTPTs come up to me and say, run their spit all the time on me. So we're trying, but I am regulated and we're trying to do the best science as possible. This is obviously safe.
I said, are your neonatal therapists actually using this information?
I get asked frequently to please run a baby's spit. Yeah. Having said that, I'm limited, right? I mean, the way our studies are written, it's not written to inform care. It's written as observational studies. So I'm learning an enormous amount. And I'm in talk with industry to potentially bring this out based on the new data coming out. So we're going fast. I don't have a great timeline. I always say if by the end of my career, I've done it, I've done it. So that's what I'm going for. But I think there's hope for others. It doesn't just have to be me doing it. I think what we're showing is there's a lot of information in saliva. And what do you want? What's your question? What do you want to see? And we're seeing that with the NAS babies. We're hopeful for the diabetic babies. And it doesn't just have to be me. So I encourage anyone who's interested you can look at saliva too.
That's great. Speaking of diabetic questions, there's those babies, we have a question about, for the study on diabetic versus non-diabetic mothers, are you controlling for different levels of mother's control over her diabetes, like mothers with A1C over 7 versus under 7?
Yeah, so we record all of that. And one of the other big issues here is obesity itself. So we know that that's another huge confounder. So this is a pilot study. These are new hypotheses coming out where we do think there's going to be disruption, and we try to control as best. But again, it's also tedious. Human studies are tedious for exactly the reasons you're bringing up. You have to wait for the right control. You have to wait for the right baby to be born. So it's a great point. And I would say those levels, sugar levels, in fact, as well as obesity levels. There is a new NIH study being launched. It's called Go Mamas, and they're going to actually be looking at mothers' glucose levels throughout gestation and then how the babies are doing after, including both their own glucose and some other clinical factors. So there are certainly people working in the field. This is an interesting cohort and it's growing. We have a lot of infants of diabetic mothers.
Thank you. How about someone would like your perspective on whether you believe it may be gene expression issues that affect full-term infants with dysphagia without other issues?
Yeah, for gene expression, sure, with the full-term babies. And I mentioned at the DNA level, so that's actually something structurally in their DNA. And I brought that up. So we think, you have a baby, you have no idea why this baby shouldn't eat. Well, we might be missing something at a DNA level. Certainly, we've had our term infants. And this is where I think such a platform could be so helpful, where we're meeting with these families and we're trying to decide, does this baby need a G-tube? How long? How long are we going to wait where we're keeping them in an environment where they're not at home, they're not with their mothers and fathers and family and being stimulated all the time? And if I could run a panel that said, look, their development is so off on all systems, maybe we could put the G-tube ball in, stop stressing the family, stop stressing the baby, get the baby home and work with the family. Because so often we're so afraid to pull that trigger because we don't know. And we think, well, if we just wait long enough, how many times have we all said that too? Tomorrow they'll eat. Maybe next week they'll eat. So we don't want to put them till surgery. But I think there are some babies, these types of platforms could be really helpful one way or the other to help guide us.
Thanks. This is a quick one. Someone just wanted to know if you could give a reminder as a researcher about what p-values are significant. So I wanted to make sure that gets out there.
Oh, p-values for significance? Yeah, sure. So just randomly, what we said is a p-value of 0.05. And that was just chosen to say, well, look, if I ran everything, the chance that this would be significant is only 5%. We've just established that as our threshold. So typically we use 0.05. When I put up the olfactory, we sat at 0.06, so it wasn't quite significant. So that may have been the driver, but usually we use 0.05 to say that we accept that's our threshold, that we believe it's significant. And, basically we would have only missed it 5%. We would be only wrong 5% of the time that it wasn't significantly different.
Another question is based off of your information and the things you're learning. Have you changed, or when do you recommend a modified swallow study at what gestation? And I know people have been complaining about that in the NICU a bit.
Yeah, I don't know that I have enough information based on what I see now, because each baby is so different. So I would continue just to recommend whatever your expertise is as you go in and see these babies and you think, is this really going to be helpful? Because those studies certainly are helpful to us, and they help guide us and help to understand what perhaps could be the problem.
Okay. I think, unfortunately, that's all we have time for questions. I want to assure everyone that if we didn't get your question, the Q&A will be posted in addition on the website as well. And I wanted to say that picture of the Big Mac and all that food is going to be stuck in my mind forever. I love the graphic. I know we all know that our infants have to feed a lot, but that graphic is going to be in my mind forever. So thank you again for this amazing work. Thank you for being here with us today, and thank you to all of our attendees. I want to just let everyone know that our next webinar will be a special webinar in February for feeding the infant with congenital heart defects in honor of Congenital Heart Defect Week. So stay tuned for more information. Everybody have a great day.
Thanks, everyone.
Bye.
Thanks for coming.